All right, you're going to have to forgive the background noise. The kids are, I got some kids taking a test, and I got some kids working on other projects, so that's just the way it's going to have to be. The endocrine system, here, what is important on this page is that you understand that there's these three components of what the endocrine system is broken down to. The fact that the endocrine system can signal near and far, the fact that what are the chemical structures of these hormones, and what the difference between water soluble and fat soluble hormones. Hormones are generalized forms of communication between tissues and cells in your body. So a hormone can have different consequences in different cells, different outcomes with different cells. So the one signal, let's take a look at the one that we just looked at, insulin. You know what? Let's look at glucagon. I think that's a better one to look at. Glucagon. In the liver, it's the same molecule, right? It's the same molecule released by the alpha cells in the pancreas. The glucagon in the liver tells it release sugar, glucose, right? In other tissue, it says it starts to change. I hate to use a triangle again, but it's going to change biomolecules to sugar. So it's the same signal, just it's going to have in different cells, it's going to have a different outcome. So it's important to understand when you're dealing with hormones that they're generalized signals that have various consequences in different cells and different tissues. Which is, by the way, one of the reasons why taking anabolic steroids to build muscle is so dangerous. If you take testosterone, doesn't just grow muscles; it does other things in other tissues. So you have three different systems, and this is incredibly important that you need to know this for sure. As far as what's important in all this, I would definitely take this as one of those that's important. You have the autocrine signaling forms of chemical signaling, and here in the autocrine system, what you need to worry about is that the chemical the cell is releasing is going to target itself. It's going to stimulate itself to do something. It's regulating itself. So there are various examples of this occurring, and we'll look at those throughout the week. So when we're looking at this across gap junctions, those cells have connections that allow them to send signals from one cell to the other. They have tunnels, if you will, that allow information to be exchanged, one cell to impact the other. This kind of gap junction signaling is done in numerous cell types that are obviously one right next to the other. The paracrine system is when you're dealing with cells that are nearby. So there's some space in between them. They may be a neuron and a muscle, neuron and muscle. There might be any number of tissues where you have to send a signal to a nearby cell to continue that effect, whatever that effect is. And then you have the cell signaling where it goes into the bloodstream and then finds a target cell. This one, of course, would be insulin would be a great example of this, or glucagon, where the pancreas creates the signal and the body cells then react to it. So that's really you need to know those three general types of signaling so that you have some idea of how cells signal each other. Here we're looking at a neurotransmitter. So this could be an axon, maybe a brain cell that runs its axon straight down. Remember, neurons you'll have to forgive my students, some are very loud. So you have neurons in the brain, but they can be very long. You have some neurons that can go down your spine and could be incredibly long. cells. So these communication, they send vesicles down through absorption, etc. And then they come down to a synaptic node. So this is an axon. This is obviously exaggerated. I shouldn't draw it this big. So you have an end to the axon, and then you have a dendrite, which is the next cells, the next neurons. Next neuronal cell. So how does this cell communicate with a nearby cell? And this being the axon of that neuron, and this being the gap, the neuronal gap. Well, let's take a look at that. A neurotransmitter is used, and the one that it was first discovered is one that's called acetylcholine. Acetylcholine is used in a number of places, usually by the way, this is showing from one neuron to the other, but you also have connections from your neuron to your muscle. When you think to move something, your neurons have to send that signal to your muscle to contract the muscle. So these neurotransmitters are held within vesicles. So this is the neurotransmitter. This is the molecule, the signal molecules held within these vesicles, and they're just waiting. A signal happens. In the case of neurons, it's an electrical potential. What that means is that there are these voltage-gated channels in the membrane of the cell, and we had discussed this earlier in the year, last semester. But you have these voltage-gated channels that open up. They open up when the potential, the concentration of ions outside, pluses, you have a lot of pluses on one side and not so many pluses on the other, then this side is more positive than this side. So in essence, this side's positive and this side's negative. If you use an acetylcholine molecule, which would be up here at the end, this is what would happen at this end of the cell, to open up the channel, then what would happen is that the ions up here, so let's go ahead and let's just use this example they have here. So as these ligand receptors bind the ligand, they open up a channel, allowing the charged ions, the potassium or sodium, through the membrane, and that depolarizes the membrane. That takes away this positive side and negative side, making them more equal, allowing for diffusion to occur and the positive charges to flow through the membrane. That depolarizes the membrane, which then propagates the signal from one end to the other. As this becomes depolarized, as this depolarization occurs here, and we call it a wave, it looks something like this. You've seen these kind of waves before on medical shows. You see that as these ions move through the membrane, this becomes depolarized, so it becomes less polar. That opens the next channel that is open because the polarization, the charge across the membranes change. This opens up, and then the ions flow through that, which depolarizes the next one, which then depolarizes the next one, which then depolarizes the next one, etc., all the way down until you get to the very end. That depolarization at the end of the neuron releases the neurotransmitter and sends the signal molecule to the ligand binding receptor on the next neuron or muscle cell, which opens up the channels, allows ions to flow through, and then that stimulates a second action potential. This wave is called an action potential. The change across the membrane, the polar charge across the membrane changes, which opens up more channels, which sends another signal down the cell. a consequence to occur. So you have a cell that's before the synapse and one that's after the synapse, and the neurotransmitter is transmitting the neuronal signal, that depolarization that happened along the neuron to the next cell via this neurotransmitter, in this case, acetylcholine. If these signal molecules stayed around forever, then you would never relax the muscle. If it's a muscle cell that's here, then you would have it contracting, it would stay contracted. That's no good. You need to get rid of these. You need to get rid of these acetylcholine signal molecules. Otherwise, you can't reset the switch. If you consider this a switch, you can't reset it. So in order to reset it, you have enzymes that destroy the neurotransmitter. So this neurotransmitter only stays in the gap for a short period of time because these enzymes exist here, getting rid of them. As you break these down, their components go back, get reabsorbed into the cell, where they're made into neurotransmitters again and put back into the vesicle, and they're waiting for a second wave of electrical potential to come down the axon and set it off again. So then you can get just this repeated polarization, depolarization, polarization, depolarization. So as you see in medical shows, you'll see a beep, beep, and that's just this repeated in EKG or ECG, and it's a horrible drawing, but you get the point. The depolarization, repolarization, depolarization, repolarization. As this signal keeps coming down, you get rid of the acetylcholine, and you wait for another signal. So when you're dealing with structures of hormones, you have some that are fat soluble. They're made from cholesterol or things like estrogens and androgens and progesterones. They tend to be steroid based, right? So they're fat soluble. Where things that are derived from amino acids tend to be water soluble. The difference is that the fat soluble molecules, signal molecules, hormones, have to be transported to the tissue, and once they get there, though, they pass right through the membrane and go right into the nucleus. So they don't really need to go through the various steps that a protein would have to deal with. So if you think of a protein's going to need, let's just call this a protein, like insulin, as an example, right? And that protein can't get through the membrane because this membrane is insoluble or water hating. So in order for this protein to impact anything inside the cell, it has to bind to a receptor that will then change its form, change its conformation, and signal in some way using a secondary signal molecule like cyclic AMP or some other secondary messenger to impact the cell. Steroid hormones don't need that necessarily. They may have a secondary messenger involved in their pathway, but the key to a steroid hormone is that it doesn't need a receptor. It can go right through the membrane and find its way right into the nucleus of the cell without too much trouble and do its impact almost fairly directly. So when you're dealing with the difference between fat-soluble hormones and proteins that are water-soluble, the difference is that water-soluble hormones like insulin need a receptor on the outside of the cell and usually use a secondary messenger to impact the inside of the cell, where a fat-soluble hormone doesn't need that, but it does need something to carry it to the cell, some kind of vesicle